This presentation will review the effects that transcranial direct current stimulation, TDCS, has on major depressive disorder, MDD. Psychological disorders have a significant physiological effect on the human body, specifically the brain. In the case of MDD, a significant amount of the physiological mechanisms are found in the prefrontal regions of the brain, particularly the prefrontal cortex. Research has shown that irregularities between the left and right dorsolateral prefrontal cortex, DLPFC, are strong indicators of MDD. Study on the imbalance between the left and right DLPFC revealed that hyperactivity in the right hemisphere was a strong indicator for severity of MDD, while reduced cerebral blood flow and metabolism in the left DLPFC was present in those with acute MDD. Research on the brain's mechanism for reward has consistently shown that reduced activity of the nucleus accumbens is present in MDD. The reward mechanism of the brain is thereby reduced, serving to increase depressive symptoms like anhedonia or the inability to find pleasure. Research is constantly underway to discover new methods of treatment for all types of illnesses. One of the latest methods of therapy for psychological disorders is TDCS, which is still undergoing improvements and testing. This method of therapy is a type of non-invasive brain stimulation, NIBS, which is quite cheap compared to other NIBSs. TDCS affects cortical excitability by increasing or inhibiting through a continuous weak current applied directly to the brain. Compared to repetitive transcranial magnetic stimulation, RTMS, the effect of TDCS is much longer lasting, with positive readings hours after stimulation. Unlike RTMS, TDCS does not cause action potentials through the breaching of the depolarization threshold. Instead, the thresholds are not breached due to the weak continuous nature of the current. This will then result in the enablement or suppression of the neuronal firing rate. TDCS has found success in the treatment of MDD. The physiological mechanisms of MDD make it perfectly suited for treatment by TDCS. The irregularities in the prefrontal cortex between the left and right DLPFC can be specifically targeted by TDCS. TDCS acts on these irregularities to reduce the hyperactivity in the right DLPFC and increase activity in the left hemisphere. Specific targeting is possible through anodal and cathodal stimulation. Dual stimulation may also be employed. Anodal TDCS is used to increase cortical excitability, while cathodal TDCS inhibits. The effectiveness of TDCS is still being heavily researched. Compared to more traditional treatments such as antidepressants, TDCS has been quite successful. A study conducted by Sergio Rigonati and colleagues found the effects of TDCS to be significantly faster than antidepressants, whilst maintaining similar effect sizes. There is still need for larger trials on TDCS. Many of the experiments on the treatment result in contrary findings. This may be due to several reasons, such as the severity of participants' depression, the asymmetrical nature of their brains, or the treatment itself. Until further research is done, it is difficult to say with certainty whether TDCS will be the future of treatment for major depressive disorder.